Good morning, everybody. It's a great pleasure to be able to present our project here in the Transformative Science section of the Gnatwork core. And so I'm going to be talking about micro-CT visualization of the parasite-host interface in small biting flies, specifically black flies and sandflies. And so we have a team of 5 co-applicants here. Bob Cheek is providing the blackfly input, Matt Rogers and Abashish Ghosh the sandfly input, and Daniel Martin Vega and myself the micro-CT techniques. So let's just start off then by having a look at, excuse me, why is it not going down? Okay, so what exactly is microcomputed tomography? Well, basically, it's just a way of using X-rays as in a clinical setting to look inside at the organs of the body. And in this case, we have a sandfly here, which is stuck between an X-ray source and a detector. And unlike in a hospital setting where the detector zooms around your body with the X-ray source, in this case here, we rotate the sandfly within the between the X-ray source and the detector. And so this is the actual CT scanner that we used, as vice versa. And you can see the X-ray source here, our sample in the middle, and then the optical magnification and a detector over on the right-hand side. And basically the output are 2D images, which can then be reconstructed as 3D images. And we'll look at that in more detail in a moment. So we're looking at 2 systems, black flies. These are Smulium damnosum, naturally infected with Onchocerca from Ghana. And then we had a laboratory model here, sandflies, Leutzomia longipalpis, and these were infected with Leishmania mexicana in London. So research questions for the black flies were, well, first of all, can we actually detect parasites in the fly vector? And a secondary question, can we compare the thickness of the peristrophic membrane between savanna and forest flies? This is a membrane surrounding the food source, which limits the movement of the parasites into the vector fly after they've been ingested. For the sandfly, again, can we detect parasites in the fly vector? And can we also detect the promastigote secretory gel that's secreted by the parasites in the gut of the fly? And thirdly, could we differentiate vectors at different stages of the infection? And of course, in addition to the science questions, we can use the images generated for public engagement. And so these images here clearly show the relatively huge volumes of blood that are ingested by these flies, and also demonstrates that we can measure the volumes of these organs. So 1.23 millimeters cubed for the black fly and slightly less for the sandfly. And we can awe the public with silly sort of facts like this. You need about 4 million black flies or almost 11 million sandflies to exsanguinate the average adult human. But we're not worried so much about what the flies are taking from us, but what they're actually leaving in us when they feed, and that's the parasites that we're looking at. So with our blackfly research, we are very pleased to have been able to work with the Council for Scientific Industrial Research, Water Research Institute in Accra, Ghana. And this is just one of the 2 breeding sites that we looked at. This is the river Tain in Ghana. It's a tributary of the Black Volta, and it's where we got our savanna flies from. So the flies were collected as pupae and were reared up to the adults. And then they were fed on cattle, which had Onchocerca nodules on them. We didn't have much success with it, actually, because difficulty of tethering the cattle, but instead we were able to feed our flies on human volunteers who were known to have onchocerciasis, so were part of the program for control. Just to show you what the sort of images, 2D images you get back from the CT scanning. So these are, each image here is taken at 90 degrees to the other. And basically you look through for the organ that you want, and in this case, the onchocerca nematode in the flight muscle. And how we visualize it in 3 dimensions is by looking at these 2D images, outlining where the organ of interest is, and then moving up several sections and outlining again. And then you can interpolate between those 2D sections and eventually build up a 3D image such as this one here. This is an onchocerca parasite. So this is a process called segmentation, and it's applied to our blackflies and sandflies. And you can end up with images like this. So you have this 2D image on the left-hand side. You can see the onchocerca in that flight muscle, and then reconstructed in 3D here. So what we've actually got is the onchocerca larva with the dark red, and the lighter red is the intramuscular space it occupies. And of course, you can then look at this in 3D and produce videos such as this one here. And the parasite is really very clearly seen in the flight muscle there. And you can visualize it in multiple different ways. Here we can just peel away the flight muscles and reveal the parasite in its intramuscular space there. And that larva, in this case, has been digitally smoothed out. So as well as being able to reveal the larva in the fly, we also were looking at this question of peristrophic membrane. And for this, we were measuring the membrane at 3 different regions of the dorsal, the anterior, and the ventral side surrounding the blood meal. It was far too thin for us to be able to asset the posterior aspect. However, most of the parasites are likely to escape out through the anterior end of the peristrophic membrane. So we were able to measure the membrane thickness, and as you can see, there was no statistically significant differences there. The hypothesis we were looking at was that the peristrophic membrane was thicker in savanna flies, and our study doesn't support that hypothesis. Okay, so leaving behind blackflies, we'll move on to our sandflies, and all this work was done in the laboratories of Matt Rogers at the London School of Hygiene and Tropical Medicine. And what Matt did was take 3-day-old sandflies and feed them either on uninfected blood or infected blood. So our controls here were obviously fed on uninfected blood, and these ones here, inf, means infected. These are the ones that were fed on Leishmania mexicana. And so we've labeled this time interval 0. This is the time of infection. 5 days later, some of the infected ones were allowed to feed on an anesthetized mouse. So they had a second non-infected blood meal. And the 5 groups that we are looking at in our CT study are shown here. So T6 denotes 6 days. This is when they were killed and scanned, 6 days after infection. And this with a plus sign shows that it had a second blood meal, non-infected blood meal. The controls were killed on day 7 after the first blood meal. We also had 9-day-old ones that only had an infected blood meal, and 9-day-old ones that also had this second blood meal. And lastly, we had some with quite a long infection, 12-day post-infection, but they didn't have the second blood meal. So you'll see these acronyms pop up associated with the images we're just going to look at. So these are the 2D images we could get of the of 4 of those different groups. We have this is the control. You can see the midgut is pretty narrow. A stomodeal valve is shut. This is the infected one. after 9 days, but it's only had the infected blood meal, and you can see the secretory gel in the midgut. The midgut's quite expanded, as is the stomodeal valve. If you additionally have a second blood meal, then you can see the additional effect that this has had. The midgut's now even wider, as is the stomodeal valve. And this is just to show you the group of flies that were longest after infection, but they didn't have a second blood meal. They also have an expanded midgut, but not quite as much as those that had the second blood meal. If you look at those in 3D, you can see quite clearly the control flies. We actually had a 9-day control fly here with very narrow guts. And perhaps the best group to look at for comparison are these here. So we have a 9-day control. This is the infected one with only one infectious blood meal. And then this had the infectious blood meal and then a second blood meal. And you can see how much more expanded the midgut is there. We also noticed that the PSG, the gel, was evident not only in the midgut, but in some specimens entered into the esophagus, the crop, and was also found in the pharynx. And if you compare here the different groups, so we had 3 flies in each group, and obviously the control ones. So this is esophagus, pharynx, crop. Red shows there's no gel in there. Green shows there is. Clearly, the controls have no gel in them. They're not infected. But those that had gel in the pharynx and crop were mostly in the ones that either had a long infection, 12 days, or this second blood meal. The 9-day infected flies that didn't have the blood meal largely had no gel into these foregut regions. So the second blood meal has a really transformative effect on the infection. We were also able to measure the opening of the stomodeal valve and the diameter of the midgut as well. Just for comparison here, the control fly, the valve was open. It was either completely shut or around about an average of 6 micrometers open. The midgut around about 70 micrometers, whereas there wasn't in this case much impact of the second non-infectious blood meal, but you can see that both the infected flies have a much more open valve and much wider midgut than the control fly. So again, these infected flies are very affected by the infection. They're flying around very bloated. Just a comparison of the volumes that we got from these flies. So this is the thoracic midgut volume, pretty low in the control flies, and also in one that's just been infected for 6 days. But the older infected flies have a more expanded midgut. But the effect of the infection itself, the gel, is even more evident when you just measure the gel itself. So in this case here, we're just measuring the thoracic gel volume. Obviously, the control flies have no gel. It's fairly small in the 6-day infection and in the 9-day infection. In fact, very similar in the 12-day, but you can see that with this second blood meal, we have a raised volume of PSG. So the flies are feeling even more bloated. You can see that just using principal components analysis, you can discriminate these different stages of infection of fly quite nicely. The controls here, these are the 6-day infected ones, and they're largely discriminated by the fact that they have a very large abdominal volume. Sorry, I should have said that, yeah, this is analysis based on the volumes of the midgut, both thoracic and abdominal. and the volumes of the PSG, and the diameters of the valve and the midgut. And this is our infected flies that had that second blood meal. Just these are the same colors as in the principal components analysis. And when you compare the thoracic PSG as a proportion of the thoracic midgut, you can see that it was much greater, almost 40% in this group with the second blood meal than in those that only had one infected meal. What was quite surprising is that it's relatively high in this group here, but that's because they actually had a smaller volume of the thoracic midgut, so the gel had a relatively bigger proportion. But even so, in these ones, it was the greatest. We weren't able to actually visualize the parasites themselves because they're so small. Our technique had a resolution of 0.9 microns, the one that we generally used anyhow. But we did scanning electron microscopy, and you can see on this infected fly here, we're looking into the pharynx, and sure enough, you can see these parasites here, which Matt's identified as leptomonad promastigotes. Okay, so going back to our research questions, were we able to answer them? With the simulium, we were able to detect parasites for sure in the vector and able to compare the thicknesses of the peristrophic membrane. The sandfly, we couldn't detect the parasites, but we could certainly detect the gel and differentiate vectors at different stages of infection. We were able to publish a paper. So far, this is on the imaging of Onchocerca in Simulium damnosum in medical and veterinary entomology. Just last week, we submitted a paper on leishmania infections plus neglected tropical diseases, and we have another couple of papers that we will be preparing and submitting in due course. And in terms of training and technology transfer, Bob was able to work with Tony and Francis in Ghana and pass on all of his knowledge there. We're very grateful to that Ghanaian team for producing the blackflies that enabled us to do this study. And we also had Debashish came across and worked in the Natural History Museum for 3 months and was able to work with Daniel and Matt and myself. He also attended the Ento 19 conference in London and was able to present a poster there on our work. So just in conclusions, micro-CT is a powerful tool for providing spatiotemporal information on the progress of infections in vectors. It gives a non-invasive, non-destructive virtual histology of any organ system, and there's really great potential for its use in a wide variety of parasite-vector interactions. And it can also produce impactful images for public engagement to raise the profile and awareness of NTDs. And I'd just like to conclude by thanking Brett and Inez at the Natural History Museum for their input, Mike in Ghana for arranging everything there, and David for his statistical advice at King's College London. And also thank you very much for your attention, for listening today. And of course, finally, just to thank the Gnatwork and BBSRC for the funding of this project.